So hi, my name is John Walsh and I'm a PhD student under the supervision of Professor James Brown, and the title of my presentation is Alzheimer's Disease in a Dish. So slide please. Thank you. So Alzheimer's disease is the most common form of dementia, a class of brain disorders which affects a new patient approximately every 3 seconds. The incidence of dementia has been increasing in recent years and is expected to continue to do so in future. In Ireland, by 2050, it's expected that over 140,000 people will be suffering from dementia. Alzheimer's disease represents around 70% of dementia cases and is characterised by the death of cells in the brain, memory loss, and disruptions to the biochemistry, structure, and gene expression of the brain. In the bottom left here, you can see a healthy brain on the left compared to the brain of a severe Alzheimer's patient on the right. Changes to the molecular mechanisms which control and regulate the healthy brain are responsible for the changes that we see in the Alzheimer's brain. These mechanisms are currently poorly understood, and because of this, current treatment methods for Alzheimer's serve to treat the symptoms of the disease rather than the actual underlying cause of the disease. So we need new and better models of Alzheimer's so that we can develop our understanding of the root causes behind the different forms of Alzheimer's to develop new and better therapies for this disease. So one type of mechanism that we've seen disrupted in the Alzheimer's brain is a reduction in the levels of a protein called TIP60. So we know that through the regulation of gene expression in the brain, TIP60 can protect the brain. And we think that by inhibiting TIP60 or disrupting its function using a drug, that we can induce Alzheimer's disease in healthy human brain cells or tissues. So we've done just that, and that's essentially creating one of these Alzheimer's models that I'm talking about. So you can see in the top right, we've done just that. I'm using an anti-TIP60 drug called TH1834. And because we couldn't get any volunteers to test a drug to see if it causes Alzheimer's disease, instead I grew mini human brain organoids, or mini brains, from stem cells. So I then treated the mini brains with our anti-TIP60 drug and analysed them to see if they had anything in common with Alzheimer's disease. And my results to date indicate that the drug treatment does mimic Alzheimer's. So we've seen a reduction in size, indicating the death of cells in the treated mini brains, which was comparable to the reduction in size and death of cells that we see in the Alzheimer's brain. As well as this, next generation sequencing analysis has shown the dysregulation of genes in the treated mini brains, which was comparable to the dysregulation of the same genes in the Alzheimer's brain. So it appears I have generated one of these new Alzheimer's disease models, which I hope can now be used to develop our understanding of the root causes behind the different forms of Alzheimer's disease and to test and develop new treatments, which could significantly improve disease outcomes as well as the quality of life of both the patients suffering from this disease and the families that care for them. Thank you very much.